Good morning. Just a small change to avoid those spammers. I've turned on uh, the subscriber only mode, right? So they won't be able to come and disturb us. All right. So welcome to Relishing Receptors Part 1. And as I said that these receptors, this topic is so beautiful topic. And in fact, you will come across multiple stories of Bhailog. Yeah, that's how I remembered. Yes, actually, none of the story, right? None of the story of any of the movie is anyway near to the stories and what is happening inside the body. The way these gangsters they work, right? कैसे एक दूसरे को they give the supari and how they transfer, right? All these things will be happening today. So let's start, right? And yes, this is part one. So gradually, I'll be taking you to all the level up to the level of pharmacology. So not only from the perspective of physiology, but from the perspective of say pharmacology or perspective of medicine, we'll be taking even some of the cases also, right? So that picture becomes crystal clear that whatever we are understanding, it has got a meaning. Bus. That's what we'll be understanding. For first 2 days, we'll be creating a very solid foundation. Today, the topic is such that try to understand every piece of it, right? Every piece of it. Once you have understood it properly, it is very much necessary to revise it properly, right? Please put the background just of anatomy. It looks anatomy. No, I'm not getting it. Okay, so right, so that's what I was talking about the receptors part. It is so interesting that I'll take you up to the level of pharmacology. And yes, we'll be talking about even the medicines also, right? That is the pharmacology, which drugs will be affecting. Then we'll also be talking about neurology, right? In neuro. And yes, we'll be taking it in reasonable depth. There would be medicine, and yes, we'll be discussing some cases, right? So this is all going to come up as we start creating our solid foundation. So this is what is going to happen as the days will progress, right? I don't know how many sessions we'll be taking for receptors, right? So that's why it has started with one. We'll go into three, so that is sure, right? More as we learn more and more concepts. Fine. We'll be starting with that point which we already know, right? And what do we know? We know about the cell membrane, right? We know about the cell membrane. And in cell membrane, in nutshell, what we got about the receptor is that this is the cell membrane, right? And now you know it very well that this is how we really draw it, right? And this upper portion, right? This portion is polar, right? And it is water loving, and in between this is what is called as the lipid loving, correct? So that's how things are going. There are some receptors which are passing through multiple times, and we call them integrated. Integrated means we just can't separate them. So these are like integrated, or there could be some channels, or there could be some receptor which are just going through one time, right? So these are one pass receptor which are related to enzyme. So basically, whether it is a 7-pass receptor, or it is some channel, or it is a 1-pass receptor, basically if we try to eliminate them, we'll be destroying. Increase the volume. Wait, I'll just do it. Now is it okay? Is it fine, Yogesh? Or I adjust it slightly more. Now it should be better. Just let me know if it is fine. Okay. So like these are all integrate. Yes, yes. Great. So these are all integrated. Try to remove, you destroy the cell membrane. And against that, there were few other types of receptors, and we call them peripheral. And when we say peripheral, that means they are attached on either side, right? So either they are out, they are, or they are in, right? And that's how they are arranged, and they can be easily separated, right? They can be separated, they can be removed with a change in the pH, change in the temperature. That's what we'll keep on watching. Now I decrease the sound. I mean, what sound is to be lowered, or it is okay? Okay, I think it should be okay now. So that's what we have understood till this point. ठीक है? Till this point. Okay. So our today's topic is now receptors, where we'll be diving more deeply into this subject, right? So receptors, which from this point onwards we'll be writing it like R, right? So this means it is receptor. Now what exactly is receptor? We learned one exact word, and that is called as ligand, right? And you remember, I know you remember this thing very well, because say coach coach hota hai. I think KKHH. I don't know whether like DDLJ, even this form, this short form is valid or not, right? But and someone added more to that this was like बहुत कुछ होता है ना? This is what you added. But truly speaking, ligand is like that, right? It is, there is some stimulus, right? That some stimulus which affects that receptor, right? Which affects that receptor, and it leads to so many changes inside the cell, right? Inside the cell. Well, bus that is a receptor, है ना? Oh, so means this short form is valid. Means you said okay, here it's valid for medicos. Chalo. So as far as we have understood, this is not only for medicos, huh? By the way, this is for everyone. Hmm. So this is what we understood, that there has to be some change. There has to be some biological change. And this biological change or biological activity or biological performance, right? These biological changes, when they occur, we say that yes, that's a receptor, and that stimulus, this stimulus, it could be anything, right? Apart from how we understood it, but it is not only that much. It's not shortcut, but it was said by me yesterday. Okay, okay, but it is good. I remembered it, so I added into my mind that next time onwards, whenever I'll be explaining this topic, I'll write that. Right? बहुत कुछ होता है. That is good thing. Okay, so maybe hormones, right? Hormones. Yes, when they act, and that's how they work, right? If there is a hormone, if it comes, that means it has to make some changes into the cell, and that's how it works. Whether it leads to increased growth, or any of the, as we see, right, or of some genetic expression, all those things, they should occur with the help of ligand. Ligand means someone who is stimulating from outside, because it cannot enter, right? So there might be some chemicals in the form, let's say we say that drugs, right? Because we'll be watching the specific drugs, that how they really work. The most important in the brain, right? Or the nerves, where the neurotransmitters, they will be working, right? Neurotransmitters. So that's how these ligands, they actually work. And when we say hormones, drugs, neurotransmitters, so in drugs, basically drugs are what? They are chemicals, right? They are chemicals. And many of the drugs, if they are given in overdose, we say toxic effects, right? Toxic effects. Yeah. So this is this is what will explain that the drug, when it is given in proper dose, yes, there is one function, but when it is overdose, when it is given more, what really happens? By the end of today's session, you will understand that it's okay to have, say, one Red Bull, but what really happens if someone takes five Red Bulls, right? What really happens? Why it is like, say, black coffee, it gives a better kick as compared to a normal apna sada coffee. Why it is said that for the weight loss, right? If you take a black coffee and then you add into that MCT, medium chain triglycerides, and that becomes what is called as the bullet coffee, right? Which shoots your metabolism to a very high level. But then all these things, they are with some side effects. So this is, you'll automatically understand, you don't have to cram really that why it all occurs. So that's how we'll be understanding these systems, right? So these are all what is called as the biological changes. They should occur with the help of ligand, right? And we'll call receptor only when it is making these changes inside the cell. We go back to our yesterday's concept, right? Because this concept would be vital for understanding everything today. One was who can cross the cell membrane, right? And we'll write it like this: cell membrane, right? That's the cell membrane. So can cross, right? One, two, three. Someone who is small, right? Someone who is small. So that means small molecular weight, right? Because they are small molecular weight. So they just sneak in, right? Small molecular weight. They can just sneak in. They can aram se andar se pass ho gaye, right? Then those who are lipid soluble, very good. Those who are lipid soluble, what's the reason? Take this particular portion, right? This particular portion, it loves lipids. So if something is lipid soluble, so it will say, okay, no problem, go ahead, right? And neutral charge, very good, excellent. The reason behind it is that this particular portion is polar, it is charged, right? Now if it is charged, right, if it is charged, so when the negative ions come, they will be repelled, thrown out, right? And when the positive ions come, they'll say, aja aja, no need to cross, you stay with me. But it is the neutral one, right? Neutral, or we call it non-polar, right? So non-polar, they will aram se, they will pass, because that polarity would be affecting the neutral ones. Very true, right? Negatively charged there, right? They are polar, right? They are polar. So when we say neutral or non-polar or non-ionized, right? These are all meaning the same, right? Non-ionized, non-ionized, right? These are the words which we'll keep on using as things appear. The reverse of this, who cannot cross, right? Cannot cross, it is pretty easy, just the reverse of it, right? The cell membrane, cell membrane. But you have to understand the reason. Large molecular weight, because there is not so much of space that such big trucks, they can really go through that small gully, right? Second, yeah, that's right, right? Second, those who are afraid of lipids, so we'll say lipophobic, lipophobic, right? And these lipophobics, they are in fact water lovers, right? Water soluble, so they cannot pass. And third, ionized. Ionized means those who are polar. Ionized, those who are charged, right? So either they will be attracted or they will be repelled, but they will not be allowed to pass through. Simple as that. So this is what we have already understood and we already know about it. One of the very interesting large molecular weights. See, all these are like peptides, right? Peptides. They are large molecules. So they will not be able to pass through that easily. So there has to be a special arrangement for them, right? Those who are polar means those who are charged. The best example is catecholamines, right? So all those catecholamines, right? Epinephrine, norepinephrine, dopamine. So they all, those which are for stress receptor, right? Stress response. So when we say epinephrine or say norepinephrine, right? Dopamine. So they all are for the stress response, right? Stress response. Basically adrenaline, right? Adrenaline. So whenever there is stress, right, it leads to increase in heart rate, right? Increase in heart rate. Why? Because cortisol, right? Increase in heart rate. Person becomes angry, right? There is anger, there is anxiety, right? Then because heart rate is increasing, so there would be increase in blood pressure. Now these things, they are okay, right? They are okay for short term when we are fighting, right? But in some cases, in many people, they are always in stress. This is the trouble. This is the trouble, right? Because it will start affecting heart, it will start affecting other organs of the body, and that's the reason there has to be a balance, right? When a person have stress, well, normal heart rate, which is said as 72 beats per minute, this is a normal heart rate, right? And 220 minus your age, and whatever the number comes, the 80% of it is the maximum one should. Yes, there is sweat also, right? Yes, there is the sweat. So this is the ideal heart rate. Anything, right? So as the heart rate is high, it is dangerous. What is the normal heart rate? Well, as discussed previously, it varies from person to person. As you exercise, as your heart becomes more efficient, your resting heart rate will start coming down, right? Your resting heart rate. The reason is the ability of the cardiac muscle to pump the blood, right? To pump the blood, that becomes more. So heart beats with a power, and that much blood is circulated into the entire body. So heart has to beat less. So resting heart rate decreases as your performance, as your athleticism increases, right? So this is very vital, because when the heart rate is low, so yes, you can work more to take it to higher levels for the better efficiency or the more exercise, right? Yes, all these things, right? All these things which are called as the sympathetic activity, right? It is called as the sympathetic activity. So there is increased alertness, and all other what we discuss: increased sweating, increased heart rate, anger, anxiety, increase in blood pressure. All those things collectively, they are like epinephrine, norepinephrine. That's what they are functioning, because it is like iodized, right? These are the polar, and as such they cannot enter. That means there has to be a very specific mechanism so that they can enter into the cell and they can create all these functions, right? All these effects. See how beautifully things have been made. Because it is ionized, it means it cannot enter by its own. There has to be a mechanism. So if we can control this mechanism, so then you can control all these activities, right? When you really learn the brain, you will find, yes, increase in alert dilates pupil. These are only few things which we have picked up. Yes, you are right, dilates pupil, because this is basically fight, fright, and flight response. Fight, fright, right, and flight response. So when we fight, right, when we fight, one of the method is, right, कि ठीक है, if we are strong तो कि मारते हैं साले को, right? But if we feel कि यार नहीं, ये सामने तो 5 लोग हैं, अपने को मार पड़ेगी, तो then we dilate our pupils कि यार भागने का रास्ता कहाँ से है, right? भागने का रास्ता कहाँ से है? So that's why dilated pupils, right? Increase alertness, because the heart pumps faster, all the organs, right? All the organs, they are getting better blood. But digestive system, no, they don't get, right? That would be compromised, because they, someone won't say that let me digest the sandwich, right, and then तेरे से लड़ता हूँ. No, right? So the reason for dilated pupils is this: fright, when the person is afraid, bhago, jaan bachao, or flight, right, higher altitudes. All these things, right, they are the fright, flight, and fight response, and that's where the adrenaline or the sympathetic activity kicks in, right? Acha. Yes, even the faster breakdown of glucose, right? So many things. The idea is that this sympathetic activity is is controlled, right? It cannot occur by itself because it is the ionized. Ionized means epinephrine, norepinephrine, they just cannot cross or the cross or enter into the cell membrane, right? So that's how the control has been given. Fine. We move on to the second one, right? Say for example, if we talk about, so this is about the ionized. If we talk about those large molecule, right? Say for example, large molecular weight structures. Now all these large molecular weight, these these sumos, right? These sumos are none but they are all peptides, right? And all these peptides, they are in reality sumos, right? Super powerful hormones, right? One of the best, insulin, right? Insulin. And it is the insulin which we'll be studying in so much detail, right? Then rest are right from the top, all the way we start, right? That is like growth hormone, then the thyroid stimulating hormone, and then all the way the lower hormones like follicle stimulating hormone in ovaries, etc., right? All those female hormones. So they are like all large molecular weight, right? So we understood large molecular weight. This means they won't be able to cross. There has to be a mechanism. That's what we really want. That insulin should not go in by its own everywhere and start controlling. No, we want to control these sumos very meticulously. That's why these mechanisms, they are there. Okay. How about smaller ones, right? All those small molecular weight, right? These small molecular weight. Now they will be able to pass pretty easily, right? They'll be able to pass pretty easily. Thyroid hormones, T3, T4, TSH, right? They will be able to pass easily. Another, this is T3, T4. I think TSH we wrote here. TSH. Only T3, T4, right? The final products. Yeah. Because once it is activated, right, so they will be leading to this T3, T4. So this is like a finished product and it will be able to pass. Inulin is small, right? It is the insulin, right? Yeah, bicarriers. Another good part. So this is like small molecular weight, right? Then those who are lipid soluble. Now see, these lipid soluble, these are almost all of them, they are the derivatives of cholesterol, right? They are the derivatives of cholesterol. And the most important of all is these sex hormones, right? So these are all lipid soluble. Yes, that is in fact extremely interesting, and even today you'll get the answer of it. That how researchers they really say they reach to this level that all these things they are occurring into the body at such a micromolecular level. That's what makes medical science so interesting, right? So here the testosterone, right? And in case of female, estrogen and progesterone, right? So estrogen and progesterone. they are all lipid soluble. One another important one, that is the vitamin D, right? It means ki in the body, if the vitamin D is at good level, it will definitely be picked up by the cells. But if the vitamin D level itself is low, nothing can be done. Same thing is true even for these hormones, right? And then if these hormones they can enter into cells so easily, then why so many complications occur? It is because these hormones they are extremely sensitive to so many other factors. So if the level itself drops, तो बेचारा cell क्या करेगा, right? When the level itself has dropped, it really leads to major changes in the body. That's why, you know, when the equity share market crashed, right? Equity share market crashed badly. Yes, there were so many psychiatric cases in which people said that they have got tremendously low sex drive or the libidos, tremendously low, right? Because they were unable to handle the stress, they were unable to handle the pressure, right? So that's the reason why it is said that keep your head cool, right? Why it is said that cold-blooded murder is considered as more dangerous as compared to someone who has killed out of anger, right? Cold-blooded murder, it means that he was in his complete senses a master planner. Yeah, by the way, anyone who has seen Freddy, right? Good movie, yeah? Just watch it, right? Means I like the movie because it is based on a book, and it's a good movie. Freddy. Yeah, I think it is on Hotstar or Netflix. I don't remember it. It is on either of these two OTP platforms. Okay, save the file. Dentist wali. But there are good twists and turns into that movie. Acha, today I'm going to tell you a story. Right? And acha, Disney Plus Hotstar pe hai. हाँ, must be. Libido means sex drive, right? It is the interest towards the opposite sex. And yes, it happens. It happens when there is big financial loss. So if the person is unable to handle that stress, and you'll be surprised. This is becoming so common. This is becoming so common, and and it is affecting even the cycles of the female, and many there are many cases in which even in their 30s, right, in their 30s, uh, female gets menopause, right? It is because of that extreme stress. It is because of, sometimes it is called as the extreme competition. बस ये करो वो करो, ठीक है, there is good to progress, but keep your head cool, right? Keep your head cool. This is so vital. This is so vital. And one day, right, let the all these topics be over, so when in physiology only it would come. So yes, we'll actually talk about all these things, right? Though from exam point of view, zero, there is no importance, but from the life's perspective, I would say million times it is powerful. It everyone must know this, because otherwise, say, as the life progresses, it actually happens. And I can give you multiple examples, right, surrounding me. Multimillionaires. But literally, it is like bus pesa pesa, it is just a number. Otherwise, they are useless, right? I can't use any other word, so but you understand it. Yeah, it is like that, right? So almost in every batch, right, whether I'm traveling, it is offline or online, I always tell to my students कि boss पैसा कमाओ, learn things properly, but know yourself so properly that after 10 years, 15 years, when you are super rich, you are enjoying the life, so then continue to enjoy that beautiful life forever, right? So it should not happen like कि then उदास होके बैठे हैं, ठीक है? Okay. अच्छा, I'll tell you a story. Right? पता नहीं कौन सी movie. Don't ask the movie. But I just remember one thing. Arshad Varsi was there, and it was some very सड़ा हुआ सा movie. Right? Sorry, Arshad Varsi, but that movie was like कुछ कोई story ही नहीं. And नहीं, today's movie is not Freddy. नहीं, today's movie is तो this movie. And actually, trust me, sometimes I really watch, I see that in IMDb rating, how is the rating, and then when the rating is say 1.2 out of 10, right? 2.2, हाँ, 3 से ज़्यादा स्टार हो, 3 से ज़्यादा रेटिंग हो तो आई डोंट वॉच दैट मूवी. आई वॉच दीज मूवीज मीन्स जस्ट टू फिगर आउट कि बॉस इतना घटिया मूवी कोई कैसे बना सकता है, राइट? मतलब दे डोंट हैव एनी सेंस. मतलब क्या घटिया स्टोरी, क्या घटिया डायलॉग्स, एंड seriously, means, but yes, at times you get something from that movie, and that was one of the movie in which, right, on this page I'll write that story only. It will take 1 minute, but that 1 minute is the concept of our entire receptor, right? Seriously, this is what really happened in that movie. So it is Arshad Varsi was the only actor which I now remember, right? So don't remember other actors, but they were pretty famous, right? All those bhai log styles. So this is Arshad Varsi and his other two friends. They were there, right? And he says कि यार चलो कहीं से 5 लाख रुपया चाहिए. तो वन ऑफ द फ्रेंड सेड कि ठीक है, एक काम कर बॉस, तो दिल्ली चला जा, मेरा एक दोस्त है, राइट, उसको 5 लाख रुपए अभी किसी को चाहिए थे, दे दिए. तो अरशद वारसी टेल्स के ठीक है मैं जाता हूँ. तो नाउ ही टेल्स दीज टू फ्रेंड्स कि अच्छा अभी तो मैं वहाँ जाऊँगा, मैं बड़ा आदमी बन जाऊँगा, तो फिर बाद में मिलेंगे, कभी कभी फ़ोन करना, right? So now he goes to that person, and these two, we just name them one and two, right? So this one and two, right? Yeah, they are in a गांव, right? And they are talking about कि यार अपना भाई अपना दोस्त गया है. Now he reaches to Delhi. Now he is some don. This is also some very famous character, but I don't remember the name. एंड ही टेल्स कि अच्छा मुझे इसने भेजा है 5 लाख रुपया. तो क्या मिल जाएगा भाई, मिल जाएगा, बस एक काम करना पड़ेगा. तो क्या, हाँ हाँ भाई हम सब काम करेंगे. कहता है ठीक है, कहता है कि ये गन उठाओ और एक मर्डर करना है. अरशद वारसी टेल्स कि अरे नहीं, ये मैं नहीं कर सकता, राइट, ये काम नहीं होगा. तो कहता है नहीं होगा, कहता है सोच लो, कहता है नहीं होगा, कहता है ठीक है. तो क्या तुमने भाई का टाइम बिगाड़ा, 30,000 रुपया दो. अरशद वारसी टेल्स, मतलब मैं 30,000 कहाँ से लाऊं? कहते कि नहीं लाया तो तुम गया. तो कहता अच्छा ठीक है, मतलब सम हाउ ही गिव्स दैट मनी एंड ही कम्स बैक टू दीज फ्रेंड्स, राइट. कहता अरे यार उसने तो मुझे लूट लिया, राइट. तो कहते कोई बात नहीं, हम दोस्त लोग हैं ना, चला जा अपने साथ, राइट. नाउ दिस डॉन, राइट, देर इज वन अनदर कैरेक्टर. I don't remember, but it was Anna or such name was there, right? He tells that okay, अभी ये पैसा इसको दो, अरे वो काम करके देगा. Now he has got a girlfriend, right, called Jia. All such strange names. Bus, this much you have to remember, right? And then there is some still bigger boss, right? He'll come into picture. I'll take the story further at that point of time, but this is entire G protein coupled receptor. The entire GPCR is like this, right? Seriously, the entire GPCR including the names and most of the things, right, they are like this. Let's learn it, right? So very classical Bhylog story. So here it is, our receptors, right? Our receptor, and in that receptor, when we said that integrated receptors, and in integrated receptors, and this one was like peripheral receptors, right? We'll be dealing with peripheral receptors later on. Today our focus is on that GPCR, G protein coupled receptors, right? We say G protein coupled receptors, which was also called as, right, it was like 1 2 3 4 5 6 7, right? So here is that stimulatory ligand, and here is where the inside cell would be affected, right? So that is also called as the 7 pass or serpentine, all same names, right? Serpentine receptors. These were like our integrated receptors. Plus in integrated we were having, so this is one. The second one which we'll be discussing, that one was like one pass receptor, right? One pass. So one pass usually will be handling all those enzymes. And the third one, right? Third one was those channels, right? Those channels. So these are because they will be sending sodium, potassium, and all those ions. So we collectively call them ion channels. Right now, we are not at all talking about peripheral receptors. Peripheral receptors will come into picture. So at this point of time, I'm just striking it off, right? I'm just striking it off so that we are not confused. We are focusing on GPCR, right? As we proceed, right, we'll keep on talking about all these systems. One point which I would like to emphasize at this point, that these ion channels, they are all controlled, right? There are various factors, right? So there will be various factors controlling channels. How? And all these factors, they have got a threshold, right? Threshold means limit. Till that point nothing happens. The moment there is one point achieved, and immediately the channel opens. So it is, channel is not like thoda open one. No, these channels are like on off buttons. On off, right? On off buttons. This is a classical system in your say houses, right? There is a relay system, relay switch, right? There is a relay switch, right? Say for example on iPhone, when we say, hey Siri, sorry, uh, nothing, thank you. So
Just to confirm, you'd like no problem. I won't. Who would you like to call?
so it is what? That means that device was sitting silently. That device was sitting silently. The moment you said one specific word, so this is like it was voice activated, right? It was voice activated. So same way, all these channels, right? All these channels, they will be, say, current activated, that is the voltage activated. That is at specific voltage, right? They will be activated. Or when you touch them, right? So they will be activated. When you press them, they will be activated. When there is change in the temperature, they will be activated. So temperature activated, right? Pressure activated, right? Pressure activated, touch activated, right? Say for example, in our intestines, there are stretch receptors. So when you stretch them, they give. That's why when we eat five samosas, five में तो कुछ नहीं होता, but if we eat more samosas, right, तो then लगता है पेट भर गया, right, because stretch receptors. But they don't have, there are no pain receptors. So if you cut them, right, there won't be any pain, right? Then obviously the all those chemicals, right? When we say chemicals, so that means those drugs, those, right, or baroreceptors, pressure receptors, very right. So all these things collectively, right, say we call them ligand or a stimulus which is actually operating it. Yes, there are, that's right. So they are spread across the entire body, right? Spread across the entire body. So that's how all those channels will be working. What GPCR is doing, 7-pass receptor, they have got their own concept, that bhai log concept, that story of bhai, which we are about to talk. And 1-pass enzyme, 1-pass receptor, or 1-pass enzyme linked receptor, seedha sada, relatively quite seedha sada. So we'll talk about this 1-pass at a later stage, right? First, let's call our chief guest, right? GPCR. When we say G protein, right? G protein coupled, right? G protein coupled receptor. Now it's not so that we are giving respect to this GPCR, right? G protein. No. It is because of guanosine, right? This G is a type of protein, that is guanosine. So that's why we are just calling it G protein, right? So here is our cell membrane. Here is our cell membrane, and this is our protein, right? This is our receptor 1, 2, 3, 4, 5, 6, and 7, right? Here is that center, right, which is picking up the signals from, yeah, bilog protein. Yeah, exactly, right? This G protein is actually like bilog protein. True. So now this 7 pass. We are dealing with right now GPCR only, right? 7 pass. Regarding others, we'll talk about. So here comes our ligand, right? So this ligand, it it is getting attached on this site, which is like an external site, right? External point. And here 1, 2, 3, 4, 5, 6, 7. All right, after passing through 7, it goes inside. And inside there is, this is an action site, right? This is an action site. The whole action, the whole movie is happening inside over here, right? Now in this inside, here is residing our G protein, right? That guanosine protein. Now this particular one is having three components, right? This is a trimeric, it is called as the trimeric protein, right? Trimeric, trimeric protein. So it has got alpha. So alpha is A, right? That is our Arshad Warsi, right? So alpha, and this is like beta and gamma, right? So that's what we keep. And initially, right, when they are in gaon, so yes, they are in contact, right? This is aram ki zindagi, everything is fine, right? And these are the components, all good. And this is what is called as the resting state, right? Resting state. Now in resting state, this this protein is in association with what is attached to it. This is important, right? So what is attached to it is GDP. It is guanosine diphosphate. Diphosphate. This particular protein loves GDP only, right? Guanosine diphosphate. So all good, things are fine, right? And this is how the life is going on, right? कोई complication नहीं, आराम की जिंदगी. But one fine day there is a message. There is a message from here, right? Someone has sent the message from outer world, right? कहीं शहर से message आया कि भाई पैसा चाहिए, right? And that message when it comes, right? So this is stimulated. When this is stimulated, the message goes inside, right? Message goes inside. It goes at the action site. There are some changes, right? And that thing is passed on to our group, right? This alpha, beta, gamma. So here, if we write it like that stimulatory ligand, right? It sends the message to that 7 pass receptor, 7 pass receptor. And then at the action site, there would be some action, correct? So we'll call this particular ligand, it could be anything. It could be some neurotransmitter, right? It could be some hormone, it could be some drug. Whosoever it is, we'll call it first messenger. First messenger, right? It is the one who is sending, who is initiating this entire process. So things go inside. Now the rest of the action is inside the cell, right? So here it is inside the cell. Now in inside the cell, it is our alpha, beta, gamma, right? And that's the whole one. Due to this stimulation from the outside, it gets that extra phosphate, and now the GDP, right? GDP is now the GTP, triphosphate. It is the triphosphate. So this GDP was the resting phase. It was the resting phase when it was having very good, say, relations with these three dost. But now with the GTP, when the GTP comes, this is what really happens. Now this Arshad Varsi tells कि अच्छा यार तेरा दोस्त है कोई जो 5 लाख देगा, कहता है हाँ मैं लेके चलता हूँ उसको, तो ठीक है अच्छा बीटा लोग, अच्छा गामा लोग, बाय बाय, राइट, मिलते हैं फिर. एंड this thing is broken. So now our this GTP and alpha, he moves further, right? And where it goes for that higher level of contacts. Interestingly, most of the medicines, maximum drugs will act here, right? That's why I'm writing it. Maximum drugs will act here. That is on G protein. G protein. Acha. One question about Bhylog you must be knowing, right? Still I'm asking. Bhylogs are good or bad? Bhylogs are good or bad? The question is that these bhylogs, they are good or bad? And once you answer it, bad, okay. Yeah, sometime good, you know? Because there was one movie of ये मुन्ना भाई, हाँ, in मुन्ना भाई MBBS, he was bhylog, right? He was bhai. Yes, if they are friend, they are good. So he said कि नहीं बापू ने बोला है, right? अभी ऐसा नहीं करने का. So that's how things go. There was one movie, that Pal 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 wala song. I don't remember the movie name. But yes, in that the bhylog was good, right? So same thing is here, right? There will be good bhylog and there will be bad bhylog. So that's the war which keeps on going, and that's the amazing war, right? Very superb. So let's see what it. KGF, KGF, KGF is a recent movie, na? है ना? Okay. So chalo, coming back to this bhylog protein, right? मुन्ना भाई MBBS उसमें ही था. No, Pal 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 wala song was. It was not Munna Bhai MBBS. It was, I think, some other song, other movie. Okay. Chalo. So this GPCR, right? Now think it like we are going bit more scientific. In that movie, that Vidya Balan was there, right? So sympathetic system. Your parents will say कि ये पढ़ा रहे हैं या मूवीज का डिस्कस कर रहे हैं. हाँ लगे रहो मुन्ना भाई, मे बी हाँ शायद. Possible. I think that was the movie, because I think she was that radio jockey, है ना? Yes, now I remember. And then he goes to the radio station. लगे रहो. अच्छा हाँ चलो अब come back यार. Else I'll start receiving emails from your parents. So so sympathetic nervous system, now we are going slightly deep. Sympathetic nervous system, when it is sympathetic nervous system, right? So that means it is all adrenaline, right? Adrenaline. And if it is adrenaline, so those receptors which are say adrenergic receptors, we'll call them adrenergic receptors. So they understand the language of adrenaline, right? Now in adrenergic receptors there are many, right? There are many. So right now we are not touching it, that what type, right? Whether it is beta 1 or beta 2, etc. No, right now there is no point in going for that classification. First we understand that what it really does, then today only we'll go for the full classification, right? Because it will take just 5 minutes, but then based on that classification, tomorrow our journey would start, when we'll take a bigger project and we'll go at an international level, right? When our bhai log from one point, now they go at an international level कि चलो भाई अब इस area में काम करते हैं, right? Then we'll go to the story of welcome, right? So that is where control, Uday control. So see all these stories. I at times their inspiration is all these Hindi no ulta Hindi movies. They are inspired by all these medical processes, right? Will feel like, and yes, actually tomorrow that welcome movie, right? That will be our nahi welcome back nahi welcome back. में तो it was not that मजेदार. It was just welcome. Welcome back was another movie. No, I think welcome. अरे इन शॉर्ट वो आलू नहीं बिका कांदा नहीं बिका वाला अरमानी सूट पहन के वो जस्ट चलता है, राइट? दैट थिंग. सीरियसली हाँ, योर पेरेंट्स आर गोइंग टू से कि यार ये क्या मूवीज चल रही है. अच्छा, so adrenergic receptor, they are those receptors who will be understanding the adrenaline, right, or the noradrenaline, and the chemical would be epinephrine, right? So epinephrine, that is our chemical, and this our chemical, this is nothing but it is ligand, right? Our ligand is epinephrine which is outside the cell, so we'll call it a first messenger. Perfect. It is our first messenger. So now the story starts, right? Story starts. So here is epinephrine. Epinephrine, right? It stimulates that 7 pass receptor. So 1, 2, 3, 4, 5, 6, 7, right? So this is a 7 pass receptor. It is stimulated. When it is stimulated inside, it generates that action, right? What's that action? Same action what we were telling, that here is our Arshadwarsi, right? Here is the beta, here is the gamma, right? And previously in the resting state it was GDP, but now it said कि boss एक extra phosphate मिल गया, so now I am with GTP, right? When the GTP comes, this is what is called as the excitatory state. Excitatory state. Now in this excitatory state, she says कि चलो let's go and do some big business, right? So here is our Arshad Warsi, right? Arshad Warsi is going to ask for some money, right? क्या मेरा नाम इतनी बार यूज किया. So anyway, from this point onwards, we'll say GTP alpha, right? And he'll say, चलो बीटा, चलो gamma, bye bye, right? अभी अपने आगे जा रहे हैं. And he will go to. So we just add this. So this is the step 1, right? Stimulates. Step 2, right? Inside effect, step 3. Excitatory step 4. GTP alpha, that is step 5, right? So now at step 5, here is this GTP and alpha. He takes to another big boss, right? Another big boss, right? And we literally write big boss, right? That cannot be, because in its reality, he is a big boss, right? And he is Anna, जो पी के बैठा है, right? And this is adenyl cyclase, right? This is one particular protein which is adenyl cyclase. This is a protein. Now this particular protein, this is where they go. Now this is a, right, he is a big boss, राइट. ही टेल्स के ठीक है मेरा काम करना है, एंड दे से सेम दैट स्टोरी कि यार मर्डर तो नहीं कर सकते, तो क्या कहते देन गो, भाग जाओ, राइट. पर यहाँ दे के जाओ कुछ. सो इन दैट प्रोसेस this GTP, right, whatever he has earned is given to this, that one phosphate, right, is given. He snatches one phosphate, and this bichara once again becomes Garib, and he becomes GDP. GDP means it is the resting phase. It is the resting phase. So in resting phase, he goes back to the gaon, right? And then alpha, and कि चलो दोस्तों वापस आ जाओ, right? And we are back to origin. And in the process, जो कमाया था, that thing is being given to this adenyl cyclase protein. He takes that. Now what he'll do? He'll say कि अच्छा this is the phosphate, अब ये पैसा है, ये इसको यूज करते हैं, right? कैसे? Go to ATP, and this is the ATP which is currency. He say कि this is the currency, convert it, right? Convert into that format which can be utilized by all those cells at a very granular level. And for that, it is what is called as the cyclic AMP. Cyclic AMP, adenosine monophosphate cyclic. This is the real value currency, right? Real value currency. So ATP is fine, ATP is generated, right? But ATP is converted to cyclic AMP, and in order to convert this cyclic AMP, you require adenyl cyclase, right? And this adenyl cyclase, as such, he is sitting silently, right? He is not working, he is not doing anything. But in the presence of this phosphate, in presence of this one phosphate, let me write it properly, just a minute, right? Let me write it properly, because this is an important step. So this is the phosphate which was Dadagiri say grabbed from this our complex, right? It is utilized, it gets activated, and it converts ATP into cyclic AMP. It is this cyclic AMP which is now called as the second messenger. It is this second messenger which has got tremendous range, right, to work at various levels. So this is now when the cyclic AMP starts rising. Now it is increasing, right? Now it is increasing. Now, this is, I am painting in green, right? It is, I am painting in green. This is where, this is like a Sharif phylog, right? But sometimes these phylogs, they have got another part also, right? We need to control them. So this red portion which I have drawn, just keep in mind it is coming soon, right? Right now it is this +1. Thick. Okay. So here is our cyclic AMP. Final result is that cyclic AMP has increased, right? Now this cyclic AMP, it acts on one more hormone. It is called as the protein kinase A. Now this protein kinase A, it will be acting on three, right? This is now stimulatory effect, right? So cyclic AMP will go and will tell. This is what ACE. ACE means enzyme, right? See how interesting things are becoming. This is the enzyme. Now this protein kinase is a versatile person, right? This protein kinase, in fact, I think let's do one thing, right? Let's make a change. Just we name this thing as Big Boss, right? And this is that green effect bus, right? And here it is, right? And here it is. This is our protein kinase ANA. Now this has effect on GIA. What is that? It has got three effects. 1, 2, 3. Now how important it is? How important? Because cyclic AMP उसने सोते हुए प्रोटीन काइनेज अन्ना को जगा दिया, and now this will say कि अच्छा अब काम करना पड़ेगा, right? So first gene, this protein kinase A, it acts on gene, and what it does is on gene it adds those phosphates, right? This is called as the process of phosphorylation. Phosphorylation. Remember this thing every time, that whenever there is phosphorylation, that means the phosphate is added, right? When the phosphate is added, right, they become happy. When they become happy, they start working, right? So this is where this protein kinase A phosphorylates gene. So this thing, it goes into the nucleus. In nucleus, we know there is DNA, right? When there is DNA, it leads to all the genomic expressions, right? So those genomic expressions, they start changing. Genomic expressions altered or the changed, right? Say I. I is what? Ion channels, right? A very beautiful effect. Ion channels. Those ion channels, they don't understand much, right? They say we just handle ions, right? Ions means all the sodium, potassium, calcium, right? So we have got specific channel. So here is a specific channel, right? Let's say this channel is for, say, calcium. If this channel is for calcium, so it will simply say ion channels, right? These channels will be opened. Why? Because of that phosphorylation, this channel will say, okay, let's start working, and the calciums will start passing through it, right? The calcium will start transferring. Again, on off switch, right? So it is what is called as the ion channels. Their state, it goes into the on state, right? They become on. So now the calcium starts moving, and this for the A, right? This we have made it for our convenience. So here we'll write enzyme, right? I know this is incorrect spelling, but that's how I remembered, right? So basically it is enzyme, right? So again, this phosphorylation, the enzyme who was sitting silently, right? This enzyme who was sitting silently, it was not doing anything. So this is one enzyme, right? And now because of phosphorylation, this enzyme is now an active enzyme. And when the enzyme is active, so that means it will make major changes into the basic body metabolism, right? So that would start increasing, right? This metabolism, it can increase the heart rate, it can increase temperature, all sorts of things. But the whole idea is how the whole process really worked, right? So it is, and all thanks to this GPCR, right? It is the sympathetic first messenger ligand, which is epinephrine. So it is red hot, right? So which is all activated, right? And that is adrenaline. Adrenergic receptors, they pick it up, right? Epinephrine received, and the signal passing through the entire one, activated at inner stage. At inner stage, right, it was sitting silently with the GDP, but it is said now, Mr. Varsi, here is you have to work, and he creates this new complex, right, with the GTP alpha complex. Beta and gamma, they are now separated. This one goes to big boss, and this is the adenyl cyclase, right? This is the adenyl cyclase. Now he grabs, he picks up, he snatches this phosphate, and then he becomes active, right? Meantime, our this one becomes garib wapas, and he goes back to his friends, and that is the resting phase. But because it is the nature, right, that nature never changes. So this, because of this, once again when the message comes, again he will leave this, these beta and gamma, again he'll come to adenyl cyclase ke bhai apne business karna hai, and again he'll take away the phosphate, and the process would continue. Meantime, this phosphate which is taken, ATP is converted to cyclic AMP, and this is the cyclic AMP which is now extremely red hot and is ready for the action, and that is what is called as the second messenger. This cyclic AMP, as the level increases, what it does, it goes to this protein kinase A, right? This protein kinase A, right? He was sleeping, but now he is awake. Now he goes to this GIA. GIA is our gene, right? Gene first. He understands only one process, the process of phosphorylation. So chalo, in the gift, give these earrings, right? So these are given phosphorylation, this gene becomes active, nucleus is active, and all the processes they start. Again, in case of ion channels, it, those ions, they start flowing because now the channels are open, right? And the enzyme, right? And the enzyme, it activates, right? This is, he himself is an enzyme. This is an enzyme, but this enzyme activates other enzymes by the process of phosphorylation. Again, the same process. And these active enzymes, they play various games into the entire body, into the body metabolism, right? This is how just one one stimulus from outside, and it has generated so many things inside the cell, right? This is one of the finest example of biological amplification, biological amplification process, biological amplification system, or the process. Yeah, cascade effect. It is like a cascade effect, right? So, but the idea is one stimulus, and it is leading to several changes, right? So, this is how things start. But say, who was the stimulator? Epinephrine, right? Epinephrine. Epinephrine is what? Epinephrine is like adrenaline, right? Just like another name. And who are they? The sympathetic one, right? Sympathetic one. What this sympathetic is doing? Sympathetic is increasing heart rate, increasing blood pressure, right? It is giving anger, it is giving anxiety, right? Always fighting mood, right? Fight, fright, all those things, dhum, 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 right? Sweating, all those things, they are going on. But well, if this thing continues, it is bad for the health, right? It is bad for the health. There has to be a control, right? There has to be a control. So now here comes the second set. Second set, we change the color. This second set is calm, cool, right? And welcome to parasympathetic system. Parasympathetic system. Parasympathetic system's other name is, the way the name of sympathetic was fight fright, right? The name of this system is rest and digest, right? Rest and digest. So cool, right? Rest and digest. And that's why when the stomach is full, right? When the not full, right? When the stomach is, means I mean full. Okay? So when the stomach is full, do we keep our eyes like this? No, right? Then the eyes are like drooping, right? Neend aati hai, right? Thanks to the parasympathetic system. So when we'll be talking about all the receptors, you will know that what parasympathetic system will be doing to our eyelids, pupils, what sympathetic system will be doing to our pupils, and why everything happens like this, because there are specific receptors. So it is just one chemical which goes, and at respective places, tuck 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 tuck, it will start operating, right? All are switches, so it will turn off, some switches on, and the final effect, right? So over here, when the parasympathetic is released, it will go and it will say pupils switch off, right? So when sympathetic system will say switch on, the pupils dilated, right? Now it will go to heart. It will to heart. It will not say कि चलो you dilate or what. It will say कि your muscle pump, right? Muscles, come on, pump fast, right? Give a powerful contraction, increase the heart rate. So it will go to SA node, okay, send the pulse fast, right? So SA node will duck 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 duck, it will start sending the pulse fast, and heart will start beating faster and faster. So that way, if we understand that these receptors, where exactly are they located and what will they do, You don't have to cram anything, it will come automatically, right? Now the parasympathetic system, very shant, cool blood, cold, right? And this will work, and for them it is called as the acetylcholine, right? From now onwards, we'll write it like this. This is acetylcholine, right? So every time we'll not write acetylcholine, it is ACH. ACH is parasympathetic. When it is parasympathetic, that means there will be parasympathetic receptors, correct? So now, and again these parasympathetic receptors, they are 7 pass receptors. But this time, they will be doing something ulta, right? They will be something ulta. These are the bilogs which will try to eliminate the effect of previous bilog, right? So that was a group 1, that was the sympathetic bilog group, and this is the parasympathetic, and that's how they both keep the entire thing in balance. Let's see how. This acetylcholine, so once again, 1, 2, 3, 4, 5, 6, 7, right? Here it is, here it is, right? And this acetylcholine, it comes over here and tells that chalo andar call lagao, right? And the message arrives, right? SMS arrives over here. Who is the G protein, right? Dodta dodta, once again, our alpha, our beta, our gamma, and that G protein, right? Which was aram se spending the nice life with GDP. This is the resting phase. Resting phase. But these G proteins, they are not the original G proteins. These are the cousins, right? These are the cousins. These are the proteins which are all very shant, right? These are the very shant, right? That is the neurotransmitter. Yes, acetylcholine is a neurotransmitter. So we'll call them inhibitory proteins. Inhibitory, because their work is chalo sab shant kar do, right? Heart rate has increased, anger has increased, anxiety has increased, sweating has increased. Come on, let's cool it down. So we'll call them inhibitory. So if we call them inhibitory, we have to go back and we have to justify this thing by telling that these were, in that case, these were all stimulatory. So these were all stimulatory, right? Not here, not here. This is not here, but here, right? They are stimulatory because they were leading to all stimulus, right? They were stimulating or the exciting all these tissues or the heart, right? The basic the body metabolism and everything was increased. So over here, this is inhibitory. Now how it will inhibit? Same process is occurring, but the final result would be slightly different, slightly completely different. So this GDP, as soon as the signal arrives, the message arrives, right? The message arrives, it will say, chalo, this GDP, same GDP, right? Now it becomes the GTP, right? GTP, and it gets attached to this alpha I inhibitory, right? This beta I and gamma I, they both are separated. गांव में ही रह जाते हैं, right? They go, and they go to that big boss, right? They go to that big boss. And who was that big boss? See, adenyl cyclase, right? This one. So this is why I told you, just remember this. So I'm putting a star, circle, and this, right? That GTP and alpha I will come here. Getting it? Adenyl cyclase is sitting in between big boss. One side, GS is coming, right? GS, that is alpha S along with GTP, right? It is telling, chalo, right? Let's work, right? And it will take away this phosphate and ATP will be active, converted to cyclic AMP and dham, all those special effects, right? Over here, the GTP and alpha I, right? GTP and alpha I, right? They come over here. That is the adenyl cyclase. And so here is that adenyl cyclase, right? Adenyl cyclase. So this adenyl cyclase, this would be inhibited because this GI is, right, this one alpha I, it is the inhibitory effect. It has got the inhibitory effect because that phosphate, right, that phosphate, but now it has got the inhibitory effect. And what would happen in this case when this thing occurs? Adenyl cyclase was converting ATP into cyclic AMP, and now this process is stopped, right? This process is stopped. When this process stops, the cyclic AMP concentration, it starts dropping. When this starts dropping, sorry, when this starts dropping, so this means the entire process, right, it slows down. So this is like a counter mechanism, counter system of what is happening in, yeah, negative feedback. Exactly, sort of negative feedback. True. Now see, this cyclic AMP, right, it is also so that cyclic AMP, once it is released, it will remain there. No, right? Cyclic AMP also gradually it gets converted to AMP, adenosine monophosphate. And this adenosine monophosphate, it is lazy. It is lazy, right? It won't work, right? Good one is for stimulation and bad one is for inhibition. Yeah, we can take it like that, right? This cyclic EMP, when it is doing all maramari, right? So then there is one control, and that is what is called as the phosphodiesterase. Again, the enzyme diesterase, so which removes this cyclic component, so it becomes AMP. This is very active, right? Cyclic AMP is very active, right? It is keeping the heart rate up, it is keeping the brain alert, right? Brain, heart rate, right? It is keeping all on high alert, right? All on high alert. But when cyclic AMP is circulating, immediately, very soon, phosphodiesterase, they come into picture and it removes, right? So once again, it is subdued. When we take tea, coffee, Red Bull, right? Something which is having all those caffeinated drinks, right? Those who are having very high level of caffeine, high level of tannin, right? They have got the habit to act on phosphodiesterase. So phosphodiesterase is now not there. If it is not there, so then this cyclic AMP survives. So it keeps on acting on the heart. It keeps on acting on the brain, and it is keeping it constantly on the alert, right? This is the reason that at night when we are feeling sleepy, right, right, so then we just have a small strong cup of coffee, and then for almost next 2-3 hours we are awake like ullu, right? जाग रहे हैं, right? Now how much is increase in the performance, that varies, but at least जाग तो रहे हैं, नींद नहीं आ रही, right? It is because your brain, your heart, right, they are firing, right, because of the presence of this cyclic AMP. They are constantly on alert because cyclic AMP has survived. Now it is not going into AMP, right? So this is how they both keep on fighting. Another point is, say this, there is another, the way we were having, say, protein kinase A, right? So what was happening, say we talked about it, right? Say I'm talking about this part. It should be crystal clear. We are talking about this part, our enzyme. Enzyme on this, what is acting? It is our protein kinase A. So we just say pKa, right? pKa. So this pKa is acting. What pKa is doing? It is phosphorylating. So it is adding those phosphates, right? And this enzyme becomes active and it leads to all those body functions, right? But then the way we have got, say, this is enzyme, right? This is enzyme and this is like an active enzyme, and this active enzyme is because of those phosphorylation, right? And this is by pKa, that is our protein kinase A, right? Protein kinase A. So it converts, but then there is another one, right? It is called as the phosphodiesterase we talked about, right? These are all phosphorylates, right? Or the phosphatase. Phosphatase. What they really do is they go over there, they remove it, right? So their function is this active enzyme now becomes inactive enzyme. Inactive enzyme. So this is how they both keep on working with each other, right? So this is like a game, right? This is literally like a game. See how it all worked. We know sympathetic and parasympathetic, right? So this is first we saw that this side is sympathetic and this side is, say, parasympathetic, right? Like a match, right? Like a match. And in this, here it is, right? We were having receptors. Which receptors? Adrenergic, right? So because adrenergic receptor, obviously adrenergic, because for sympathetic nervous system there is adrenaline, right? That is epinephrine, norepinephrine, etc. In parasympathetic we were having acetylcholine, right? So we'll call them cholinergic. ठीक है? In cholinergic again there is muscarinic and nicotinic. That's what we are coming to that, right? But first right now we just understand, say, cholinergic. Cholinergic means those receptors which are sensitive to acetylcholine, period. Correct? So this is one thing. In this, what was our G protein, right? Our G protein, especially the alpha one, right? The alpha one, right? They were all the G stimulatory, right? They were G stimulatory because they were going to that adenyl cyclase and they were telling adenyl cyclase, bhai, convert AMP to CMP, अपना एक phosphate ले लो, right? That's what they were doing. While in this case, they were G inhibitory, right? G inhibitory. Again, they were going to that adenyl cyclase, but then that was phosphodiesterase when it was telling cyclic AMP back to AMP, right? नहीं काम करना काम, right? So this is where, right? Here it is, phosphodiesterase, right? This phosphodiesterase, when it is inhibited, right, and it was keeping cyclic AMP to AMP, right? So that was, so here we have got adenyl cyclase, right, which was converting AMP, right, AMP to CMP, and over here it is this adenyl cyclase, right? It has got the inhibitory effect, so that we have got again AMP back, right? AMP back. There was protein kinase A, right? Protein kinase A, and what it was doing? Phosphorylation, right? Phosphorylation. Phosphorylation was what? Phosphorylation was adding those phosphates. And when those phosphates they were getting added, so then enzyme was becoming active, right? Enzyme was becoming active. And then obviously there are all various functions. Over here, just the reverse, right? That activated enzyme by phosphatase, right? Phosphatase. So these enzymes, right, they are removed and the enzyme becomes inactive. So the process stops. Enzyme becomes inactive. That's the contrast which keeps on going non-stop, right? It keeps on working, it keeps on going one after the another. Now there are some other types also, but what we are doing is we are not going into that detail. We'll see it tomorrow, right? Just adding a very important, very nice classification, and this will improve the whole concept, that what we said about adrenergic, what we said about, say, cholinergic, right? We'll see that okay, there are so many divisions, right? What they really do, that will keep on learning step by step. This is the piece of information which I'm just giving you, right? It is completely fine if you forget it, because but exposure to this is important, right? So here is our all GPCRs in the entire body, all the GPCRs. Yes, all of them, they are in synapse. Synapse is what? Say neurotransmitters, right? Say all those neurotransmitters, right? They go like this. So this is the synapse. So in between, that's how they release. We'll understand this process also, right? That what really happens when the stimulus comes, how those vesicles, they are broken when they release this, and then there is how the recirculation of this enzyme, this neurotransmitter occurs, right? But yes, all these things, they are occurring into the synapse. Very right. Coming back to this, all GPCRs, right? That is what we are discussing. First one, right? What we say is for, let's say, for parasympathetic, right? Parasympathetic. When it is for parasympathetic, we'll call this thing as cholinergic, right? Cholinergic. Do remember this, cholinergic. Why cholinergic? Because it is ACH, acetylcholine, right? Acetylcholine. This acetylcholine, that's why the receptors will be cholinergic receptors. These cholinergic receptors, there are of two types, right? There are two types. They act at different area. Their function is different. This is what will be, yes, synaptic laughter, yeah. So this is what we'll be discussing when we'll actually discuss the parasympathetic, right? Yes, we'll be discussing in this week only, but this is for understanding. Two types, one which is called as the nicotinic, the effect which is similar to as if the nicotine, how it is acting, right? Nicotine, so it is called as the nicotinic effect. And the second one, it is called as the muscarinic effect. Muscarinic, right? Muscarinic effect. So we have got cholinergic receptors, but now we have nicotinic receptor and we have got muscarinic receptors, correct? These nicotinic receptors, they are ion channels, right? They are ion channels, correct? And these muscarinic effects, they are like 7-pass receptors, the GPCRs, right? These ion channels or the nicotinic receptors, they are NM and NN, right? NM or NN. And while muscarinic ones, they are M1, M2, M3, M4, M5, right? Very simple. Just don't confuse at all that what they will be doing. That is what we'll be discussing. But you should just know that these are the types, right? Nicotinic and muscarinic bus, right? And they are collectively cholinergic receptors. ठीक? The second group should be of sympathetic. Sympathetic. When we say sympathetic, that means they all will be related to adrenaline. So we'll call it as adrenergic receptors, right? These adrenergic receptors, you have to remember it like, say, KISS, not KI, but KIQISS. But still, some things you cannot remember with Sharafat, right? And this way you remember it very instantly. Okay, so 1, 2, 3, 4, 5. Order is important, that's why, right? So we'll call it alpha 1, alpha 2, beta 1, beta 2, beta 3. So it means we'll call this as adrenergic alpha 1 receptor, adrenergic beta 1 receptor, adrenergic beta 3 receptors. It means they will be at various levels, various points. Now see beta 1, beta 2, beta 3, all they are GS, GS, GS. Now you know it. GS means what? They all are stimulatory. Correct? They are stimulatory. It means they will be increasing adenyl cyclase. ठीक? Adenyl cyclase, that big boss, right? When that thing would increase, obviously it will lead to increase in cyclic AMP, right? When the cyclic AMP is increased, yes, there would be increase in even the protein kinase A. And when the protein kinase A is increased, obviously it will affect GR, right? So it will affect genes, it will affect our ion channels, and it will affect enzymes, right? So all of them, they will rise, right? Stimulatory effect. The alpha 2 is very specific, right? It is very specific because it will, in fact, this alpha 2, it is GI. So it's not so, remember, it's not so that when it is adrenergic, so then everything is high, right? Some things are even subdued. Say for example, you want to tell, okay, digestive system, abhi, now we are fighting, so you just stay silent, right? So you need to say some structures not to work and some structures to work, right? So that's why there is inhibitory component which is also needed. So if it is GI, so then there is decrease in cyclic AMP, right? You know the reason now very well. And that's how they will be acting on, right? So here, when it is inhibitory effect, right? So it leads to, just remember, it will lead to potassium efflux, right? How it occurs, we'll discuss. But just potassium efflux. E means exit. Exit means it goes out, right? Potassium goes out. When the potassium goes out, potassium will go out from the cell. The cell will become negative, right? Cell will become negative. Acha. So this is for the potassium efflux. And this alpha 1, this is very special. It is called as the GQ. Now this GQ, it is completely different. It has got its own style, right? So this would be the topic for tomorrow. It is what is called as the PIP2, right? And then there are two parts of it. I'm just writing, you keep in mind, right? Tomorrow you will get the exact answer that what it will really do, right? So this is what is called as the IP3. These are all, and its function would be it increases calcium, right? Increases calcium, especially say in heart muscles. When the calcium comes, and that's where the contraction starts. And the daga, right? This is also a Don's name, right? And this is what will do protein kinase, right? Till this point, we were having that protein kinase A. Now this is our protein kinase C. And this was Q, I, S, S. Make it more intense. S, right? Yeah, phosphatidylinositol. That's what will be. But this is where we add one more S, right? So this is alpha 1, this one is alpha 2, this one is beta 1, this one is beta 2, this one is beta 3, right? So that is how these sympathetic systems, they are classified. Then the third one, now all are pretty easy. The third one, which is called as the dopaminergic, right? Our catecholamines. So dopamine. So from dopamine, dopaminergic, right? These are the dopaminergic receptors. Those receptors which are sensitive to dopamine. So in that case, ligand is dopamine. So they are divided into simple, just D1 and the D2. This D1 is GS, stimulatory, right? When it is stimulatory, so obviously it will increase cyclic AMP, right? Makes sense. And when it is, say, inhibitory, so it is GI, right? And it would decrease cyclic AMP. And the last one is what is called as the purinergic, right? Purinergic receptors. Purinergic receptors. So purinergic receptors, these are also called as the purinoceptors, right? Purinergic receptors combine the word, so purinoceptors. Now we know that there are four major proteins, right? Adenosine, guanosine, cytosine, and thymine. So purines and the pyrimidines, right? These are called as purines. So we have got adenosine, right? And by the way, these purines, they are having two carbon rings, right? So I just say 2C, or let's write it properly, right? Two carbon rings. So they are purines. So adenosine and guanosine. And over here, these are all called as the pyrimidines. And in pyrimidines, there is one carbon chain, right? And they will be part of cytosine and thymine. So cytosine and thymine. We are focusing on only this group. So that is purinergic receptors. These are the receptors. Its importance is very high, right? Because purinergic receptors, they will be playing a major role in learning, your memory, right? Your memory, then your movements, locomotion, right? Movements, your sleep, which is very vital, right? Sleep, because you must have seen that disturbed sleep, right? Then your behavior, so that is the behavior pattern. These purinergic receptors, they play big role in this, right? So these purinergic receptors, when we say, so purinergic receptors, they are divided into two parts. One, what is called as the P1, and second is P2, right? So good. This P1 is simple apna GPCR, right? GPCR. This one is different. This is P2Y and this one is P2X, right? And this P2Y is once again GPCR. So that means those 7-pass receptors. It means it is sensitive to that ATP, ADP, right? All those things will come. P2X, that is special. It is ion channels. These are ion channels, right? So those who are sensitive to GDP, right? And, say this GPCR. So this means that for them ATP, right, that will play a major role. While in this ion channels, yes, here also ATP will play a role, but process changes, right? Method changes. So these are like basic classifications of all the types of receptors, right? So we have got 4 types of receptors. Parasympathetic, that is what we call as the cholinergic. Then we have got, these are main, cholinergic and adrenergic, these are main, right? In this, we were having nicotinic and muscarinic, and over here, adrenergic, it is alpha 1, alpha 2, beta 1, beta 2, beta 3, is QI double triple S. And then finally dopaminergic and the purinergic. Right? Dopaminergic and purinergic, they are specific for the specific purpose. Most of the things, they will be games will be played between cholinergic and the adrenergic receptors. Right? We stop at this point for today. Tomorrow we'll be starting. Tomorrow we'll be going for a very high speed, quick revision of this portion, right? Some portion of this, and then we'll take it to the next level. Yes, we'll revise for today also, right? So that is how we shall proceed. So let's go for a very high speed revision. This is like a superb story, and here it is. Right, so cell membrane integrated, and it has got say 7 pass receptors, it has got channels, and it has got 1 pass receptor, and 1 pass means it is enzyme linked or the enzyme related. Well, this is polar, and in between is lipid loving lipid. So receptors, we have got the ligand, it acts on the receptor, and then there is some change in the inside. And these ligand could be hormones, drugs, chemicals, toxins, neurotransmitters, etc. Both the process must be balanced. If any one of them behaves abnormally, it can be fatal because it has the direct effect on cardiovascular. Very right, very right. This is the reason that when it said if the sympathetic system is constantly up, right? If sympathetic system, so few people they are angry on everything, right? For no reason, for no reason, but unnecessarily. When the person becomes angry, he is actually harming himself more as compared to someone else. Yes, definitely, definitely in Alzheimer's. Yes, it affects. Right. So that's why the balance is needed. So concept, small molecular weight, lipid soluble and neutral or non-polar or non-ionized one can pass through cell membrane, right? This and this. Cannot cross, well, large cannot cross, lipophobic cannot cross, and ionized cannot cross, right? Sympathetic, it is for alertness, increased sweat, increased heart rate, anger, anxiety, blood pressure, etc. Right? Fight, flight, and fright response, and the resting heart rate, right? That is a crucial one. Large molecular weight, insulin, growth hormone, thyroid stimulating, follicle stimulating, luteinizing hormone, right? Prolactin, right? All those hormones, right? Vital hormones. All of them, they are peptides and they are of large molecular weight. Small molecular weight, T3, T4, the finished product, right? Lipid soluble, these are the derivatives of cholesterol, and they enter straight away, right? Sex hormone, testosterone, estrogen, progesterone, vitamin D, all of them, right? That's why their level should be maintained properly, right? Our bilog story, and here is our receptor, integrated and peripheral, something which we have not touched today, right? We'll take it tomorrow. This is integrated, 3 types, GPCR 7 pass or serpentine, and the second type, which is 1 pass enzyme, and the third type, which is the ion channels, right? There are factors controlling these channels. They act like a relay switch, on off switch. When that threshold is reached, tack, right? That channel opens and things start moving, right? These channels, they can be controlled or operated by either the voltage, changes in the voltage, right? As the voltage changes, they start opening. Change in the temperature, change in the pressure, touch, chemical, all these are collectively called as a ligand, which will make some change inside the cell at the external stimulus. GPCR, guanosine, protein coupled receptors, and that's like a new name which we have given to them is bilog protein. Bilog protein, its real sense, right? Here is, right, a call comes from outside and it tells कि message भेजो अंदर, message भेजा, this is at the action site, this G protein, right? This G protein, which is like a stimulatory protein. What to do about pathology? It sickness reducing. Pathology, ठीक है. We'll see. We'll start even because just a minute. Let's finish this and then we'll talk about it, Anand. Okay, just 2 minutes. So this is like stimular, right? So alpha, beta, and this is the GDP, and in the life was good, resting phase, trimeric protein, right? Everything fine. and then the message comes. When the message comes, right, action site, and he tells that chalo now it is GTP with our alpha, and it goes for the bigger business, leaving the friends behind, beta and gamma. Maximum drugs they are acting over here, that is the G protein. Sympathetic, right, when it is like epinephrine, he sent the message from outside, so that's why it is called as the first messenger, right, that ligand it is called as the first messenger, and that's the adrenergic receptor which is working on. And here it is, this is the first messenger, works, and this 7-pass receptor takes the message inside. When it is inside, from the resting state GDP, it becomes into excitatory state, that is GTP. GTP leaves this beta and gamma on one side, and GTP and this Arshad Warsi, they go for the big business. They go to the big boss. Big boss tells ke murder karo. He tells ke no nahi karenge to ke theek hai lao phosphate lao. That's the भाई से भाई का टाइम खराब करने का चार्ज, right? And this becomes वापस बेचारा गरीब. He goes back and meets with his old friend, and they go into the resting phase. Meantime, this adenyl cyclase becomes happy, right? इसको तगड़ा माल मिला. So then he becomes happy and he says कि चलो let's start doing the work. ATP is converted to cyclic AMP, right? Cyclic AMP is something which is real value currency. So this cyclic AMP, which is also called as the second messenger, cyclic AMP, it is given to still bigger boss, right? Protein kinase Anna, right? Protein kinase A, right? He is stimulated. ये सोते हुए को जगाया. This enzyme is activated, right? And this is like Gia, right? Where he acts. G is for gene. Gene is where the phosphorylation, that is the phosphorylation, that is the phosphate bond is added, and this gene becomes active in the nucleus, and it is called as the genomic expression, right? Whenever we'll be using the word genomic expression, that means there would be some change in this entire sequence. And you know, when there is change in the sequence, it would lead to changes in some processes in the body, some growth, right? All those processes. These are the ion channels. Ion channels, they are switched on, and the moment they are switched on, if it is for calcium, the calcium starts moving. If it is for magnesium, the magnesium starts moving. So tomorrow, when we'll be understanding those pumps, tomorrow when we'll be understanding about the action potential, right, at that point we'll see that these channels, right, they work so effectively, tack, right, on, and the whole bunch, those ions, they start moving inside, and inside the environment is a calcium aya chalo contract, because they will be getting those actin myosin filament to wait hi kar rahe the, right? They were waiting कि chalo let's start the contraction process. So that's how so many things would is going to occur. So these enzymes, once again, this one enzyme activates another enzyme, and that's how the basic body metabolism, it goes to a higher level, right? So this is what is called as the biological amplification system, right? When there is one stimulus, but effect is multiple fold, right? Multiple fold or multiple times. Epinephrine, adrenaline, that is sympathetic, increase heart rate, increase blood pressure, anger, anxiety, sweating, right? All good. Someone to counter them, that is parasympathetic. So that's why it is called as rest and digest, aram, right? Relax. So here it is, that's why blue color, right? It is a night time, and acetylcholine enters, sends a message silently, ki chalo chup chup, right? Message. And this is the alpha inhibitory. Once again, they were resting, but then this is the new group which is formed. Once again, this group has got habit to go to that big boss, right? Adenyl cyclase, but he tells ki bas chup, you will not work, right? Because it has got the inhibitory effect. That inhibitory effect will stop ATP to getting converted to cyclic AMP. Cyclic AMP was actually the real value currency which is to be utilized by all of them. Now this is not there, right? So it is said, okay, band. This cyclic AMP to AMP, right? That is the reverse one. It is by phosphodiesterase. So this is this phosphodiesterase hormone, right? This phosphodiesterase hormone will convert cyclic AMP, which is active, to AMP, which is lazy, right? Which is not working. So that is the phosphodiesterase enzyme. This phosphodiesterase enzyme is inhibited by tea, coffee, all those Red Bull, caffeines, etc. So when cyclic AMP was there, things were active. Now not, right? It becomes lazy. Similarly, enzyme protein kinase A, the counter which drugs are responsible for this process? So many, so many. There is a huge list, right? We'll talk about the drugs tomorrow. So enzyme, and this becomes an active enzyme, but phosphatase, right? It stops this process, right? And it becomes an active enzyme, right? So that's how things go. Adrenergic receptors, cholinergic receptors, GS to GI, adenyl cyclase, right? Cyclic EMP is converted to cyclic AMP is converted to cyclic EMP, and protein kinase A, because of phosphorylation, right? Because of this effect on enzyme becomes active. Here it becomes ulta. Because of phosphatase, it takes away those phosphates and it becomes inactive, right? All GPCRs, parasympathetic, they are also called as the cholinergic, right? Parasympathetics, they are called as the cholinergic, remember. So we have got the nicotinic receptors and we have got the muscarinic receptors. Nicotinic, they are ion channels, right? On off, right? The two types, NM and NN. GPCRs, M1, M2, M3, M4, M5, right? All of them, they are having very specific purposes, right? Regarding the sympathetic, right? Regarding the sympathetic means they are adrenergic receptors, right? Adrenergic receptors. Remember QI triple S, right? Pronunciation is same, right? So that's why easy to remember. This is alpha 1, alpha 2, beta 1, beta 2, beta 3. So just remember GQ, right? So this is BIP2, IP3 and DAG in detail tomorrow, right? Alpha 2, this is GI inhibitory, so there is decreased cyclic EMP. Beta 1, 2, 3, they all are stimulatory. That means there would be increase in the adenyl cyclase, there would be increase in the cyclic AMP, and there would be increase in the protein kinase A. Rest are dopaminergic, right, which is D1 and D2, and one is stimulatory, second is inhibitory, right? And finally, this is the purinergic, right, worth discussing because it is for learning, memory, movement, sleep, behavior pattern, etc. Right? And these are purines and pyrimidines. That is, AG is purine and CT is pyrimidine. So purinergic, right? They are P1 and this is P2. That's how we divide. P1 is GPCR, P2Y is GPCR, P2X is ion channels. If you remember this much, right, it will make the entire concept extremely easy when we'll be discussing that what would really happen when some specific say medicines they will be acting on it, right? Say for example the medicines which are acting against something. So we have got, let's say, right, just one example and then things will be much better. Say over here, muscarinic effect. Muscarinic is what? Parasympathetic, right? Parasympathetic. So if we have got muscarinic, right, listen carefully, we have got muscarinic antagonist, right? There are agonist and antagonist. Antagonist means it is going against it, right? So what this will do, it will do, because this is muscarinic antagonist, right? So it will selectively inhibit muscarinic receptors, right? It will inhibit muscarinic receptors. It won't touch nicotinic receptor because this is a specific drug, right? We'll come to drug. But muscarinic antagonist, a drug which we say that okay, it is muscarinic antagonist, that means it will be acting not on the entire system, right? Not on the entire parasympathetic system. It will affect only the muscarinic receptors. Now these muscarinic receptors, where they are present, right? It is like, say, in the parietal cells, right? In the parietal cells where stomach, right? In the parietal cells. These parietal cells, they are doing what? Well, they are releasing acid, right? Acid secretion. They are doing acid secretion. Makes sense, na? Makes sense. Because these parietal cells, they are having muscarinic receptors, and these muscarinic receptors, because they are parasympathetic, that means rest and digest. So when you are resting, when you are digesting, right, so then these parasympathetic receptors, that is the muscarinic receptor, will say कि chalo release acid or shuru karo acid digestion, right, acid secretion. So let's start the digestion. But we are having acidity, right, or we want to stop this secretion. So what we'll do, right, we'll come up with some medicine which will say कि अच्छा, stop these receptors so that they don't release acid, right. We want to cut down the gastric acid secretion, right. So if we say pyrenzipine, pyrenzipine or pyrazipine, right, both are correct, it will selectively inhibit muscarinic receptor. This would be blocked. When this is blocked, obviously acid secretion would drop. This is one of the example that how it really acts, right? Because it would straight away go for that. Acha, one more very interesting. Parasympathetic, right? Parasympathetic is doing what? Parasympathetic is, say, pupils, right? Pupils will be constricted, right, because we are now in the sleep mode. So pupils will be constricted. You want to dilate it, you want to dilate these pupils. So you must have seen ophthalmologists, they say okay, put atropine drops, right, atropine drops. These atropine drops, they will be acting, they are again muscarinic antagonist, right, they are muscarinic antagonist. They'll say that okay, you are constricting the pupil, let me restrict you, pupils, they don't have choice but to dilate, right, they will dilate because things which were getting constricted due to that receptor, we have blocked that only, right. So now pupil will dilate. So this is how we'll be having agonist and antagonist, right? There will be few blockers and there will be few which will be stimulating, right? So that's what, as we'll proceed, we'll talk about all these receptors, we'll talk about all these agonist, antagonist, because for say someone who is negative, right, someone who is negative, but if you inhibit this negative, so it becomes positive. You're right, right? Or say if the heart rate is increasing, right, heart rate is increasing, so heart rate increases because of the sympathetic, right, sympathetic system, it is increasing. So if we go for sympathetic antagonist, so it will take the heart rate down, correct. So that is how we'll be understanding the entire body, all the transmitters, all the receptors, and how they really work. This is the difference where what we were talking about over here, that why there are so many alpha 1, alpha 2, beta 1, beta 2, beta 3, because some are present in heart, some are present in lungs, some are present. So if we figure out a drug which is exactly acting only on beta 1 of adrenergic receptor, so that means that whosoever is having beta 1, only they will be affected, rest will not be affected. So that way we have got very specific effect on the human body, minimal side effect. Obviously if we take something like a wide adrenergic receptor, so it will affect alpha 1, alpha 2, beta 1, beta 2, beta 3. Our purpose was just for this much. Unnecessarily it is acting on other areas, and which is leading to so many complications, right, right. So this is how things will go. Acha, what was your question? If we give agonist of muscle, obviously that's right, because if we give agonist, that means it will selectively, it will tell to parietal cells ke chalo go ahead, right. Antagonist, right, that will decrease. So simply, you are supporting the digestive system, right? You are supporting the digestive system by agonist, right? Okay, yes, go ahead, right? Stimulus. So obviously it will lead to increase, correct? And if you are telling a antagonist, right? So ulta. So you are, though it is digesting, you'll say, no, don't do it, right? Selectively parietal cells blocked. So fine, they will, there would be decreased. Right? So thank you so much. And tomorrow we'll take the things from this point onwards. Right? But trust me, by the end of this receptors thing, literally you might have to revise 2 3 times, but then the concepts will be so clean, and all the way it will be helpful to you till the end, right? At every level in medical, right? So thank you so much, right? I appreciate your patience, seriously, right? Thank you, and see you tomorrow, right? Bye bye. File saved. Within few minutes you'll be getting a link in the description. Thank you. That's right. That's right. You are right, DK. So see, this is what really happens. The moment you start understanding the process, instantly it becomes crystal clear. Okay, this is what they are doing. Thank you, and see you tomorrow.